Well, then let's move on into our final lecture of the cardiovascular system, not the final lecture of the unit, but the final cardiovascular lecture. We'll be talking about our blood. Most cells of the human body cannot move around to obtain oxygen and nutrients or to eliminate carbon dioxide and other waste, and instead these needs are met by two fluids, blood and interstitial fluid. We've talked at great lengths this semester about interstitial fluid, all the way even back to the very first lecture of the semester. We learned that interstitial fluid is fluid between cells, so we can say it bathes our body cells. It's constantly renewed by our blood, and that's because water and stuff dissolved in our water can leave the blood, becoming interstitial fluid, or it can go from the interstitial space into cells and then back out into the interstitial space. So we just have a constant circulation going on. Blood itself is a liquid connective tissue composed of a liquid extracellular matrix, which we also mentioned in our very first lecture this semester, called blood plasma, that dissolves and suspends various cells and cell fragments. Okay. Once again, I want to reiterate that fluids are always circulating. That includes blood and interstitial fluid. So substances constantly are moving, diffusing from the blood into the interstitial fluid and from interstitial fluid into the blood, and so on and so on and so on and so on. Overall, we can say that our blood has three major functions. Its primary job is centered around transportation, but it also protects us and it helps regulate various things. It transports a lot of different things: oxygen from our lungs to our body's tissues, carbon dioxide from those same tissues to the lungs to be exhaled out. Nutrients are picked up from the digestive tract, mainly from the small intestine. Metabolic wastes are carried to the kidneys to be filtered out and made into urine. Hormones, they're carried to target cells, which we've previously talked a lot about. Stem cells are transported from bone marrow to tissues. And we can actually transport heat as well to regulate body temperature. It sounds kind of weird because heat is not a substance, it's a form of energy. So how can we transport heat? And well, think about your blood like a glass of water. Okay, glasses of water can change temperature, right? Okay, water actually can hold on to heat. Okay? And as we're about to find out, about half of your blood is water, so your blood can hold heat. Okay. And by controlling where our blood is, that can affect your overall body temperature. Okay. We've talked at great lengths this semester about what we do when we get cold and what we do when we get hot. We shiver and we sweat, right? Okay. But we also reroute blood. Okay. When we're hot, we want to increase the flow of blood to our skin. That explains why a lot of times if you're hot, your face turns really red. And that's because you're flushing the skin with blood. That way it's closer to the surface of the body where that sweat is to cool the blood internally. And then, we also can like fan our face, or maybe there's air blowing. That will also cool the blood, which can cool your overall body temperature off. The opposite occurs when we get cold. We kind of try to prevent blood from flowing to the skin. That's why sometimes you look a lot more pale when you're really cold. You're keeping that blood more internally, so it's less likely to lose its heat. Okay. Protective mechanisms of the blood, a lot of which centers around our white blood cells and how they provide immunity. We'll learn about those later in the lecture, as well as in our next lecture. We'll talk in our next lecture more about inflammation, which is something that happens anytime a tissue is damaged, whether it's from a cut, from a burn, whether it's from a broken bone, it doesn't really matter. We always have inflammation associated with tissue damage. We'll talk a little bit here, but in our next lecture, a lot more about antibodies and what they do to neutralize toxins and pathogens. Okay, and then we'll talk about blood clotting using our platelets, actually more so in this lecture. And then we regulate things using our blood, like the fluid distribution, by moving fluids across our capillaries. And we have proteins in our blood that help buffer and stabilize pH of our body. So important. All right, just to kind of introduce blood, I want to talk about what makes it up and what's in it, how much we have and whatnot. We can describe blood as being denser and slightly more viscous. So there's that word viscous or viscosity that I was talking about in our last lecture. So it's a little thicker than water and has a slight sticky feel to it. Its temperature is around 38 degrees Celsius or 100.4 degrees Fahrenheit, which is about a degree Celsius higher than what you'd find like inside your mouth or inside the rectum. Has a slight alkaline pH of 7.35 to 7.45. We like to see around 7.4, which if you think back to when we talked about pH, beginning of the semester, we learned that's that 0 to 14 scale where 7.0 is in the middle, and that's like, pure water where it's neutral. Okay, just above that is 7.4, and that therefore makes it a base. And it's a very weak base at that, but it is a base nonetheless. The color of your blood will vary with its oxygen content. If it's rich with oxygen, it's going to appear more bright red. If it's oxygen poor, despite what people think, it's not blue, it's actually dark red. And what you might see, like, for example, if you look in the back of your hand or like in your legs, you might find vessels that are appearing blue. It's all about refraction of light and nothing to do with the color of your blood inside of those vessels. And healthy young females will have about 4 to 5 liters of blood, which you think about is like 2.5 big pop bottles full. Males will have about 5 to 6 liters. So 2.5 to 3 liters, or 3 of those pop bottles. That represents about 8% of our overall total body mass. Okay, and looking at the next page, 2 main components make up whole blood. Okay, 55% roughly is our plasma, and the other 45% will be our formed elements. Okay, plasma is the liquid portion of our blood. It's the extracellular matrix. Okay, and it is predominantly made up of water, about 91.5% of it, which is why I said about half of your blood is water. Okay, over half of your blood is plasma, but most of that is water. So when you kind of factor things in, it's almost 50% water. I don't know what the actual percentage was. I've never done the calculation, but it's pretty close to half water. Okay. The rest includes various stuff that's dissolved in that water, most of which are 3 major types of plasma proteins: albumins, globulins, fibrinogen, and then you've got some other proteins like hormones. You've got electrolytes, you got nutrients, gases, waste products, so on and so forth. Okay, we'll talk a little bit more about those in just a minute. Okay, formed elements are going to have a visible structure and represent the cells and cell fragments, constitute the other 45% of our total blood volume. Okay, it includes red blood cells, which are the most numerous cells in our blood. In fact, these are the most numerous cells in our body. It's kind of misleading here because it says 4.8 to 5.4 million. But if you look down at the bottom, it says in parentheses numbers per, and that's short for microliters. Okay, so that's not in those 4 to 5 to 6 liters. That's a microliter. If you don't know, okay, think about it this way: microliter is a very small fraction of a liter, and that's because if you want to sample blood, you wouldn't want to take a whole liter out of that individual. Okay, that'd be like 20% of their blood. So you take a microliter, okay, and according to what I Googled, how much is a microliter of blood? Okay, what's 1/1,000,000th of a liter? Okay, and that is extremely, extremely, extremely small. Okay, it is less than a drop of blood. In fact, a single drop of blood is going to be roughly 35 microliters. So very, very small fraction of blood. And there's like 25 or 35 trillion red blood cells total in the body. So way more than what you see listed here. You've got 5 different types of white blood cells, each of which is different in number and different in function, as we'll talk later about. And then platelets are not actually cells, but they're formed when little pieces of a cell break free from the bigger cell called a megakaryocyte. We'll talk about those later on in the lecture as well. Before we talk about the cells, though, let's talk a little bit more about the plasma, okay, which I've already kind of described the stuff here at the top, but it's a mixture of water, around 91.5%, and then about 8.5% of it is dissolved solutes, includes proteins, nutrients, electrolytes, nitrogenous waste, hormones, and gases. Of these components, plasma proteins are the most abundant, totaling 6 to 9 G/DL. Okay, this G/DL is short for grams. That's what the G stands for. per DL is short for deciliter. And that's just another fraction of a liter. Okay. And so we're dealing with a very small amount. I want to say like a deciliter is like not quite a cup, like a measuring cup. Okay. That's how much blood is. So it's quite a bit more than a microliter, but it's still not a whole lot. Okay. But you're going to find in that small fraction of blood 6 to 9 grams of these plasma proteins, most of which are synthesized in our liver by hepatocytes and include the following 3 major categories. Okay, albumins, globulins, and fibrinogen. Albumins are going to serve to transport various plasma solutes and help maintain osmotic and pH balance of our blood plasma. So they act like an Uber in that they help carry stuff in our blood, but then they're also involved with osmotic balance, which is like your water levels, and then pH balance, which obviously is related to acids and bases. Globulins, they are an essential group of proteins, and there's actually 3 different types: alphas, betas, and gammas. Alphas and betas are similar to albumins, and I didn't even really list them off. That's okay. They are going to be transporters, and that's why they're like albumins. Gamma globulins we talk a lot more about because they are what we commonly call our antibodies. Okay? Proteins that are produced during certain immune responses. Okay, in response to exposure to foreign substances, or what we call antigens, which you'll see that word a lot here in this lecture and in the next. Things like bacteria and viruses, okay, are what we commonly call an antigen. When we encounter those, we will produce millions of different antibodies that are capable of binding to it, and then eventually disabling it. We'll, like I said, talk more about those in our next lecture. There's 5 different types of antibodies. We classify them as like IgAs, IgGs, so on and so forth. The IG is short for immunoglobulins. This is just like a fancier word for antibodies. And then the final major protein in our blood is fibrinogen, which we'll talk a lot about in this lecture when we talk later about forming blood clots. It's the precursor of fibrin, which is a sticky protein that forms the framework for blood clots, which is what we do whenever we bleed. So looking here on the next page, table 19.1 lists off the components in our plasma. Obviously at the top is water, then the 3 plasma proteins that we just talked about. But then it also lists off the other things that I kind of just mentioned and didn't really get into. Gives you a nice description of what they do for the blood, but not going to really get into specifics there for you. Over on the left side, if you don't know, this is not pee, but this is actually a bag of plasma, which can be donated if you aren't aware. You find plasma centers a lot of times in college towns because you get paid for it. And those centers like to rely on college kids that are typically pretty broke. It's a pretty cool process how it all works. Essentially, they stick a line in your arm and that pulls the blood out just like you would if you were donating whole blood, which I think we are donating here. There's a blood drive next Thursday, sometime next week here in Atchison. But they pull the blood out, and instead of collecting the whole blood, what they do is they run the blood into a machine that separates the cells, the formed elements from the plasma. The formed elements, all those blood cells get put right back in, so the line pushes that blood back into the body while the plasma collects in the bag. And for that reason, you can actually donate plasma like a couple of times a week, whereas with like whole blood, I think it's like every 6 or 8 weeks, could be even longer. And that's what it's kind of sounding like off the top of my head. It's a lot harder to reproduce your blood cells than it is to reproduce your plasma, because as we just mentioned, most of your plasma is water, and that's pretty easily replaced. But it's a very beneficial thing for both parties. Obviously, the people that donate it get paid, but plasma can be used for medicines and a lot of other things. So, very, very nice process. All right, with that said, we're ready now to move on from the plasma to the cells of our blood. Okay, first thing I want to talk about is how we produce them. Although some white blood cells have a lifetime measured in years, most of them are going to last only hours, days, or weeks, meaning that we need to continually replace them. A negative feedback is going to help regulate the total number of red blood cells and platelets in our circulation, and therefore will hopefully keep their numbers typically steady, where there's not going to be too much of a fluctuation, although that does occur from time to time, which is very different than what we see for white blood cells, because the number of those will vary in response to challenges by invading pathogens and other foreign antigens. For example, you have some of your white blood cells that are more specific to fight off against things like viruses, whereas others are more specific to fight off against bacteria, or some are going to target like parasitic worms that can, invade us, which sounds really gross, but that does happen. So would you expect, for example, your bacterial specializers to go high in number if you are sick with COVID? No, because COVID is a virus, right? So you'd expect the viral targeters to go up in those conditions, like COVID or like chicken pox, and the bacterial numbers to actually stay lower, and the bacterial fighters, I should say. And that's actually a very useful way that doctors can try to figure out what's wrong with an individual or a patient. If they're not aware, they can test their blood and see which white blood cell numbers are up, which ones are down, and that can give them a good heads up as to maybe it's a virus, maybe it's a bacteria. So we'll talk a little bit more about that later in the lecture. Process in which we form the elements or formed elements of the blood is called hemo or hematopoiesis. Both those terms mean the same thing. Before birth, it begins in the yolk sac of an embryo, and this is all very, very early in fetal development, before any organs are developed. It continues in the fetus in their liver, spleen, thymus, and lymph nodes. But just before birth, in the last trimester or so, the red bone marrow becomes that primary site. And I would highlight this is the continued site or source of blood cells throughout our life. So in our last trimester through the end of our life, our red bone marrow is where we produce our blood cells. Red bone marrow can be described as a highly vascularized tissue, so it gets a lot of blood flowing to it. Okay, and that makes sense because this is where we make the blood cells. So obviously it's very tied in with the blood. Within our red bone marrow are specialized cells called pluripotent or multipotent stem cells, which also go by the term hemocytoblasts. And actually, if we jump ahead to the next page, we can see one of those at the top. Okay. That multipotent stem cell or pluripotent stem cell has the capacity or capability to develop into many different types of cells. These cells reproduce themselves, proliferate, and differentiate into all of our blood cells. Okay, that includes the red blood cells, the platelets, and then all of these here on the right side of the page. These are different white blood cells: eosinophils, basophils, neutrophils, monocytes. And then these three over here are all lymphocytes, even though this one's not labeled as such. It's just natural killer cell. It is a lymphocyte. Okay. They also can form other cells like this mast cell here, which is in our connective tissues. Monocytes can transform and become big cells called macrophages. B cells or B lymphocytes can form plasmacytes or plasma cells, as we'll sometimes see them listed, which is the cells that actually produce our antibodies. Okay, so we can form a lot of different things: blood cells, macrophages, even reticular cells that were pictured there. Those are found in places like your lymph nodes, mast cells, which were shown there, adipocytes, which are fat cells, and some can even form osteoblasts in our bone, chondroblasts in our cartilage, and muscle cells, obviously, in our muscle. So, in other words, this cell here at the top of the next page is a really great starting point for making just a whole bunch of other types of cells. Okay. Now, when it comes to those blood cells, once they're produced in the marrow, they must enter the bloodstream through what we call sinusoids, which are those capillaries I showed you from my anatomy lecture that look like Swiss cheese, where they got these large holes or sinuses in them. And in order to form our blood cells, those pluripotent or multipotent stem cells produce two further types of stem cells, either myeloids or lymphoid stem cells. Oops, wrong way. The myeloid stem cells begin their development in the red bone marrow and give rise to red blood cells, platelets, monocytes, neutrophils, eosinophils, basophils. So in other words, myeloid stem cells forms all of our blood cells with the exception of lymphocytes. And that's because lymphocytes come from lymphoid stem cells, which hopefully kind of makes sense. Lymphocytes, lymphoid stem cells. Another big difference between lymphoids and myeloids is that lymphoids begin their development in red bone marrow, but don't complete it until they make their way into like lymphatic tissues, like our lymph nodes, for example. And now, during hemopoiesis, some of the myeloid stem cells can differentiate into progenitor cells, while others develop into precursor cells. If we look here, we kind of have them coded with, green and kind of this pinkish shade. I guess that's a pink, maybe it's like a light purple. Okay. Progenitor cells give rise to more specific elements of the blood, whereas precursor cells, which always end with blast at the end of the name, they develop into actual formed elements of blood after several cell divisions. Notice all of these, like I said, end with blast. Sometimes it's directly from that. Sometimes there's another cell in between. Okay. Like in the case of this reticulocyte, in the case of this megakaryocyte. But we ultimately end up with a red blood cell or platelets from it. As far as the exam goes, I'm not going to expect you to, be able to regurgitate all this stuff. Make sure you know what the process of blood cell production is. Hemo or hematopoiesis involves negative feedback, where it takes place, which are red bone marrow. I just maybe know like the major highlights. Okay, we'll start by talking about red blood cells, or what we would more fancily call our erythrocytes, which are the cells in our blood most responsible for carrying oxygen from our lungs to our tissues and carbon dioxide from our tissues to the lungs. Okay, an individual red blood cell or erythrocyte is a discoid shaped cell, approximately 8 microns in diameter and 2 microns thick. We look at this next page, we can see that in its dimensions labeled at the bottom. It's a very small cell. It's among the smallest cells in the body. And it is a pretty unique cell in that it has this very circular shape, but more importantly, it's unique in that it's got this kind of indented feature to it. And the reason that it's indented the way that it is because it actually does not have a nucleus anymore. Okay, if I go back to this illustration, the reticulocyte that you see here is the precursor cell to forming it. And it is a cell that actually ejects its nucleus. And that's a purposeful thing. And that's what triggers it to kind of indent and cave in on itself like it is shown at the bottom. Now, why would it want to do that? Well, then it's more of an empty cell, and so that it can have more of this inside. More hemoglobin, about 33% of the cytoplasm of a red blood cell is made up of hemoglobin, which accounts for about 280 million molecules of hemoglobin per cell. This is the molecule that is most responsible for transporting our oxygen and carbon dioxide. Something around 98.5% of the oxygen that's carried in our blood is carried by hemoglobin. So by far and away, the majority of our oxygen is carried by hemoglobin. It does carry carbon dioxide as well, but only like 23% of your CO2 is carried by hemoglobin. And we'll talk more about that here in a couple of weeks when we get to our respiratory lecture. So hemoglobin is obviously very involved with oxygen and carbon dioxide transport, but it's also going to play a role in regulating blood flow and blood pressure. It does so by releasing a hormone called nitric oxide, a gaseous hormone that, keep in mind, is not produced by hemoglobin. It's actually produced by the endothelial cells that are on the inside of our blood vessels. Okay, but that hormone produced by those cells binds to hemoglobin. And so under certain circumstances, it can release that nitric oxide, which is going to be a very potent vasodilator of our blood vessels. So it's going to help increase blood flow through that tissue. If we actually think back to the very last lecture that we just finished talking about, and this was among the very last things we talked about on Tuesday, we were talking about autoregulation, and we learned about vasodilating and vasoconstricting chemicals. Nitric oxide was one of those vasodilating chemicals. There is another protein in our red blood cells, aside from hemoglobin, that's very important. And this is the enzyme carbonic anhydrase. It's found where not only in our red blood cells, I should say, but also other cells of the body that we'll talk about here in the next few weeks. But it catalyzes a reaction joining carbon dioxide, CO2, and water forming carbonic acid, which then dissociates into bicarbonate ions. Okay, we talked about this process the other day. Remember, CO2 and water form this acid, and I brought that up because that was all part of what we call the chemoreceptor reflex that we were talking about with blood pressure. Okay. Like I said, carbonic anhydrase is an enzyme that's not just found in red blood cells. So we'll be talking about it again here in the next couple class periods. Wonderful thing about forming this bicarbonate is that bicarbonate ions are very, very great at helping us maintain normal blood pH levels. So we'll talk about what we call the bicarbonate buffer system here in a future lecture or two. Okay, let's talk a little bit more about hemoglobin. Okay, I've described it as a protein, which is true to a degree, but it's also a little bit misleading because it's part protein, part not protein. Okay, the protein part of hemoglobin is what we see here with the blue squiggly lines, which might look like it's just one long blue squiggly line, but in fact is actually 4 blue squiggly lines. Okay, those are the globin part of hemoglobin. Okay, and there are 4 chains. 2 of them are betas, 2 of them are alpha chains. Okay. Associated with each one of those chains is a non-protein structure called heme. Okay, and each of those things that to me always looks like a flying saucer or a UFO is an iron-containing heme, like we show here in Figure C, and we describe it as being a heme with iron in it or containing iron, because at the center of it is a ferrous iron Fe2+, which is very important because this is where 1 molecule of oxygen can bind. And therefore, since there are 4 of these per hemoglobin, that means 1 molecule of hemoglobin can transport 4 oxygen molecules. And if you wanted to be kind of crazy, you could take that 4 molecules of oxygen, multiply that by 280 million hemoglobin molecules for each red blood cell. So over 1 billion oxygen molecules can be carried by 1 red blood cell. Then you multiply that by the trillions of red blood cells in our body, which I say, and I do this because I want to demonstrate just how amazingly great our blood is at having the capacity to carry oxygen. Okay. Once again, hemoglobin does transport carbon dioxide, but it's not nearly as important. It's only about 23%, whereas it's like 98.5, 99% of our oxygen. Okay, the life cycle of a red blood cell is fairly short, only about 120 days. And a lot of that is because they get pretty beat up. Okay, there's so many, and they are squeezing through very small and tight spaces, especially in our capillaries, that it's fairly easy for them to get, worn down. Okay, and unfortunately, as they wear down, they can't repair themselves because they don't contain a nucleus and other organelles. Therefore, they cannot synthesize new components to replace the damaged ones. So instead, they get fragile with age and are more likely to burst as they move through narrow places like in our spleen. So we remove them from circulation and we destroy them using phagocytic macrophages, which also takes place in the spleen, but also the liver. The great thing is it's not kind of a wasteful thing because we are able to recycle some of the breakdown products, especially when it comes to hemoglobin. And the globin and the heme portions of hemoglobin become split apart. Remember, globin is the protein, and making up proteins are amino acids. So essentially, you can take globin, break it down into those amino acids, and then those, as you can see there, can be reused to synthesize new proteins. Maybe it's going to make new globin, maybe it's going to make an entirely different protein, but we get to use them for something else nonetheless. Okay. When it comes to heme, we remove the iron and we can recycle that. We can see that taking place right there. Whereas the rest of the heme really isn't as useful to us. And so we're actually just going to get it out of the body using our feces and our urine to excrete it out. Okay. What we see here taking place in the liver is the movement of bilirubin, which is what's formed from that 9 iron heme. Some of that goes into the small intestine and will be converted into stercobilin, which is actually where the brown color to feces comes from. So if you ever wonder why your poop is brown, now you know. The other avenue is to get it out through our pee in the form of urobilin, which is what causes our pee to be yellow. So probably stuff you never really cared to know, but now you do. The reason that your pee is yellow and your poop is brown is because of what happens to the hemoglobin in our body. Now, earlier I said that red blood cell numbers, along with platelet numbers, should stay fairly steady, although that is possibly not going to always be the case. So identifying and reporting the numbers of red blood cells and hemoglobin in the body might be useful to try to figure out what's wrong with an individual. Probably the most commonly used test is a blood hematocrit, which is where they just look at the percentage of whole blood volume composed of red blood cells. In men, it's about 40 to 54%. In women, it's about 38 to 46%. So it's a little bit lower in women than it is in men, most of which is due to the hormone testosterone that is present in higher concentrations in men. Stimulates the synthesis of a hormone called erythropoietin, which is the hormone that we'll talk about in just a second, that triggers red blood cell production. And so looking at the three test tubes over here on the right, we are illustrating a condition normal, and then one where there's too little red blood cells and one where there's too many. So which of those is just right and what we would want to see as a normal condition, A, B, or C? A. Okay. B would be described as being an individual with anemia, which is a term we've probably heard of before. Typically, people that are anemic, what would you describe them as being? What do people commonly call anemia? Low levels of, well, iron. Okay, that's what I was looking for. People that say if they're low in iron that they're anemic, it's not always 100% accurate. If you are low in iron, you're probably going to be anemic, but the true definition of anemia doesn't necessarily relate to low iron. You don't have to be low in iron to be anemic. It could be just because you don't have enough red blood cells because you are bleeding too much. You have hemophilia or something like that. We'll talk a little bit more about that at the end of the lecture. And whereas like figure C here, we're showing what's called polycythemia, okay, where there's too many red blood cells. We talked about that the other day. That makes the blood more viscous or thicker. Okay. Polycythemia is not a natural thing, but it can happen. Like if somebody does blood doping, where they remove blood from their body and then naturally reproduce it and then pump that blood back into the body right before they go and compete in a competition. Very commonly done by athletes wanting to gain an advantage. You see it a lot in like cycling sports and that kind of stuff. But that's polycythemia on the right. Hemoglobin concentration is a mass of hemoglobin per volume of whole blood. Typically, we want to see it around 15 grams per deciliter, so 12 to 18 is normal. And then probably not use a whole lot. It's just red blood cell count, which is the number of red blood cells per volume of whole blood. And that's what we saw in that figure a few slides ago, 4.2 to 6.2 million red blood cells per microliter. So it's actually a little bit different as far as the number. I said, what, 4.2 to 6.2. So here it's 4.8 to 5.4. I guess the values I give are a little bit more encompassing or a broader range. Not that that's really that important as far as numbers. I'm not going to ask you to know that. Okay, so earlier we learned that production of blood cells, all the blood cells, is a process called hemo or hematopoiesis. Well, now we have a similar sounding word, erythropoiesis, which is specifically the production of our red blood cells. And we call it erythropoiesis because the other term for a red blood cell is an erythrocyte. Okay. It begins with a process where we form a precursor cell called a proerythroblast. And this is all once again taking place in the red bone marrow. Okay, a proerythroblast is one of those precursor cells, which if I go back to this illustration again, we can see right here. That's the cell that comes before what's called a reticular cell, or excuse me, a reticulocyte. Okay, these precursor cells, known as proerythroblasts, differentiate through cell divisions until they eventually form a reticulocyte. Okay, which are cells that have ejected their nucleus and causes the cell to indent, giving it its distinctive shape. Okay. Now, those reticulocytes that are formed will leave the bone marrow, where they enter the circulation by squeezing through the endothelial cells or between them and making their way into our blood capillaries. And then it takes about a day or two for them to mature into a fully functioning red blood cell. Okay. As we learned earlier, production of red blood cells is regulated through negative feedback. And a lot of that is related to a decrease in oxygen delivery to our kidneys, and in some cases other tissues. Okay. What we'd commonly call the condition hypoxia, which is one of those other terms we learned the other day, decreased O2 content in the blood, which we talked about when we were discussing the chemoreceptor reflex. Okay. So in our figure on the right, we show some type of stimulus disrupting homeostasis by decreasing oxygen delivery to the kidneys, okay, where the kidney cells then detect that low oxygen and therefore increase the synthesis and secretion of erythropoietin or EPO into the blood. Okay, EPO, short for erythropoietin, is what triggers those three steps where we go from the proerythroblast eventually to larger numbers of red blood cells. And therefore, if we have more red blood cells, we can increase the delivery of oxygen to our tissues, and our control condition can be, fixed, and we can, properly deliver oxygen to the kidneys. So what can cause us to decrease the amount of oxygen delivered to our kidneys? Okay, for example, exercise is something that it's not a very long-term cause of it, but when you exercise, remember we learned that whenever that sympathetic nervous system is active, we decrease the flow of blood to places like our kidneys and our digestive organs, and therefore that decreased flow of blood to the kidneys and our liver is detected as low oxygen availability. Okay, going to high altitude, like climbing a mountain or just going up into places like Breckenridge there in Colorado, reduces the amount of oxygen in the blood. It's not actually because what most people think that there's less oxygen in the air. That's not actually true. It's all about pressure of oxygen, which we'll talk about later in a few weeks. And then a loss of lung tissue or function means that you're probably not going to get enough oxygen in the blood. That can cause hypoxia as well. Okay. Now I mentioned that people that can experience this letter C here called polycythemia can occur because of blood doping. You can actually get your hands on synthetic erythropoietin, and that's going to essentially do the same thing, where you don't have to take the blood out of the body. You can just trigger the production of more red blood cells, and therefore end up with more ability to carry oxygen to your working skeletal muscles. So that's another thing that a lot of athletes do, which is in most committees and or athletic organizations, like I know the Olympics committee, the IOC, they test for EPO, synthetic EPO, if those are elevated, and they test for blood doping. Those things are illegal. Something that you can do as an athlete that's not illegal is live in certain places in the world, okay, on purpose. If you live at a high altitude, you're essentially going to trigger the production of red blood cells, okay, and that's why people that want to gain an advantage might go and live in the mountains and train there. The bad thing about training at a higher altitude, you typically can't exercise as hard, so it actually might cause you to get out of shape a little bit. So some athletes actually invest in like hyperbaric chambers where they can sleep in them and mimic high altitude without actually living there. It's kind of crazy, but pretty effective. All right, white blood cells, fancy term for those are leukocytes, formed elements in our blood, providing us protection against infectious microorganisms and other pathogens. They are very different from white blood cells in that they retain their organelles throughout their life, therefore show a much more complex internal structure. So they do have a nucleus, they do have lysosomes and other organelles. What they do not have is hemoglobin. Okay. Now we classify them into two categories based on essentially what they look like under a microscope. It's either the presence or the absence of these granules that either kind of show up when you stain them, which is what you have to do when you want to view them under a microscope. And the preparation of a slide, you put a little stain on there. They're going to either look kind of speckly, like they have freckles, or kind of grainy, which is why we call them the granulocytes or granular leukocytes. Or they're going to look not so grainy. They're going to be called the agranulocytes or agranular leukocytes. Remember, if you see the letter A at the beginning of a word, it means the opposite or not. So the agranulocytes are the non-grainy looking ones. We'll talk about those next, but first let's talk about the three granulocytes. Granular leukocytes or granulocytes are the ones that look like they have freckles. And it's because they have lysosomes and other membrane-bound organelles that appear as conspicuous colored granules when stained in a blood film. There are three granulocytes, and if you haven't already seen it, noticed, they all end with the same suffix. The ending phils or phil, if you're only talking about one, is common between these three, which is not the case for what we would call the agranulocytes, which are going to end very differently with the suffix site or sites at the end of their term. Okay. So those are the five types. Let's talk about each one in greater detail, starting with eosinophils, which are going to combat the effects of histamine and allergic reactions. Can also phagocytize antigen antibody complexes, and they can destroy certain parasitic worms. Basophils are going to liberate, or in other words, release heparin, histamine, and serotonin, which are chemicals used in allergic reactions that intensify the overall inflammatory response. These guys function very similar to what we call mast cells, which are found in our connective tissue, because they release the same chemicals involved with inflammation. Now, we'll talk a little bit more about inflammation, like I said, in our next lecture, as well as those chemicals and what they do for inflammatory responses. But I always tell students, think of basophils and mast cells as like some of the most annoying cells that we have, because of the fact that they release things like histamine, which is what causes a lot of allergy symptoms. Being itchy, having, runny nose, typical things that we experience, whether it's, being allergic to a cat like I am, or if you're like allergic to pollen, you're releasing histamine, which explains why people that are trying to alleviate those symptoms might take what they call an antihistamine. Ever heard that phrase before? It's a common thing in allergy medicines like Claritin and whatnot. All right, then you have neutrophils, which I would describe, and I would highlight their description that they are the first leukocytes to arrive at an infection site. In other words, these guys are your first responders. And I would also highlight that they are very capable of performing phagocytosis, which if you just want to kind of visualize, think of neutrophils. There's two different types of these guys that can do this. The other we'll talk about on the next page. But they run around like Pac-Man and gobble stuff up. Things like viruses and bacteria, which are pathogens, and digest them by releasing chemicals that destroy it. Chemicals like lysozyme, strong oxidants, and defensins, which we'll kind of describe those in just a second. All right, the other two cells of our that are in the white blood cell category are the agranulocytes, which include lymphocytes and monocytes. Lymphocytes, and I would highlight this, are the major soldiers of your immune system, and therefore we'll talk a lot more about these in our next lecture. There's actually several size differences when it comes to lymphocytes between small and large. Physiologically, there's not a whole big discussion there. There's really No big difference as far as we know, but it is still useful in clinical settings to try to figure out what's wrong with somebody. The three major functional types of lymphocytes are the T's and the B's, and then the natural killer cells. Okay, the T's and the B's we will talk a lot more about next lecture because they provide us with our most sophisticated forms of immunity. Okay, T cells are responsible for attacking viruses, fungi, transplanted cells, cancer cells, and some bacteria. Unfortunately, they are going to have a side effect of causing transfusion reactions, allergies, and rejecting transplanted organs. So in other words, if a person has, like, for example, a kidney transplant or a heart transplant, they would actually have to take special drugs that would decrease the activity of their T cells so that their body doesn't just destroy that organ that they've just been given. Okay. B cells or B lymphocytes are particularly effective in destroying bacteria and in activating their toxins. Okay. You want to kind of great, easy illustration and explanation of what they're able to do. Let me just kind of do something really quick. So you got B cells. They can form, and we'll talk, like I said, I keep saying this, but we'll discuss this more in our next lecture. They can form what we call memory B cells. Or they can form plasma cells. And the importance of that, well, actually, memory Bs can eventually become plasma cells or form them, kind of essentially. But the importance of plasma cells is they are what we actually use to form our antibodies. And then that's how B cells provide us with immunity against things like viruses and whatnot. And as you can see, when we see the word memory on this page, okay, that's a very important thing about your B's and your T's. They have a memory component, which means what? Any idea? They can remember things. Okay. Not like remember, facts and like the number of bones that we have, but remember something they've encountered before. Okay. They make up what we call our adaptive immune system, and therefore enable us to have like a really quick reaction to something that we've encountered a prior time. And in other words, these guys, are involved in our why we get vaccines, which we'll talk more about vaccines in the next lecture. Natural killer cells, which have a really cool name, and they sound like secret agents to me, but they're not quite as complicated as the B's and T's. They do, though, attack a wide variety of infectious microbes and certain spontaneously arising tumor cells. And then monocytes are blood vessel or blood cells that can leave the blood. And then once they do, they can enlarge and differentiate into our biggest Pac-Man cells of the body, what we call macrophages, which can stay put in a tissue. We'd call those fixed, or they can wander around. And so we call those wandering, where they can then gather at sites of infection or inflammation. They do take longer to reach a site of infection than neutrophils, but they come in larger numbers and destroy more microbes. So neutrophils and macrophages are the two cells, like I said, if you want to kind of visualize them, they help us go through phagocytosis. Now, how do those macrophages and neutrophils know where to go? How do they know to target a specific area where you might find a virus or some bacteria? Okay. Well, that's what we'll talk about here. Unfortunately, because our body is not 100% sealed off, it's inevitable that we will have viruses, bacteria, fungi, toxins invading deep tissues. Okay. Our mucous membranes are exposed to the exterior, like the inside of our mouth, and then our bronchial tree is a very easy way for stuff to come in. Same thing with our nose, even our eyes, our ears, the other ends of our body. Those are open. We're not a closed organism. Okay, and then our skin is not 100% formidable either. It's easy to cut through your skin. So we're constantly and continuously exposing our body to microbes and toxins. And eventually some of them are going to get into our body and cause diseases. In order to combat these microbes and toxins, white blood cells must leave the bloodstream and collect at sites of pathogen invasion or inflammation. Because, think of it this way, viruses and bacteria are going to be in other places besides the blood, which means that those white blood cells must leave the blood eventually. Okay. They do so in a process called emigration with an E. It's a pretty cool process where they roll along the endothelial lining of blood vessels, then they stick to it, and then they are able to squeeze between the endothelial cells to get out of the blood. Now, this is kind of unique because typically, we'll talk more about this in our next couple slides into the next lecture, but most of the time, it's hard for stuff to stick to the wall of our blood vessels because they're kind of like a non-stick pan. They don't want stuff sticking to their interiors because of a lining that they have called prostacyclin. But in the case of white blood cells, they can stick to that wall. Especially is important for our neutrophils and macrophages, which are the active phagocytes of the body, process where they ingest bacteria and dispose of it, okay, and other dead matter. Okay. Then they release chemicals that are going to engulf or that are going to destroy the pathogens. That includes lysozyme, an antibacterial enzyme, which is actually similar to antibacterial enzyme, and strong oxidants that are similar to household bleach. And the neutrophils also contain defensins, which are proteins that exhibit a broad range of antibiotic activity against bacteria and fungi. So how do those phagocytes know where to go? Well, I always use this analogy, and it works because I experience it from time to time. If you've ever been at home, whether you were doing the baking or your significant other or somebody else in your house was doing the baking, and whether you're making brownies or a cake or cookies, that smell of that deliciousness kind of permeates throughout the house. Okay, and like in our house, when my wife makes her brownies or whatever, pretty soon that me and my boys are going to end up in the kitchen because we get hungry. We're chemically attracted to the kitchen. Well, our white blood cells can't smell, but they can almost like taste. Okay, when we have bacteria, when we have viruses, when our body is damaged, these various chemicals called chemoattractants are released throughout the body. And then those white blood cells can detect that. It's almost like they taste it. Then they go to where that's coming from. That process is called chemotaxis. And we'll, like I said, talk more about this. I keep telling you this more in our next lecture, which is why we talk about these things back to back, because they're so intertwined. Now, earlier I said that white blood cell numbers can fluctuate depending on what's going on in the body. Okay. An increase in the number of circulating white blood cells usually indicates that something's wrong. It indicates inflammation or some type of infection. We call this leukocytosis. Things like invading microbes, strenuous exercise, anesthesia, and surgery are very commonly experienced causes of leukocytosis. Okay. And earlier I mentioned that a doctor will examine an individual's white blood cell numbers in order to probably figure out whether it's a virus, a bacteria, whether it's a fungus, whether it's something else. Okay, that test is called a differential white blood cell count, where they just count up each of the 5 different types of white blood cells to detect infection or inflammation, determine the effects of possible poisoning by chemicals, drugs, monitor blood disorders like leukemia and the effects of chemotherapy, or detect allergic reactions and parasitic infections. Okay, So the lot can be determined by just counting the number of red blood cells. For example, we look at the table over on the right. A high number of neutrophils is common if an individual's suffering from a bacterial infection, whereas if they have a high number of lymphocytes, it's because of a viral infection. Okay. If you've ever seen the TV show House, it's almost every episode that I've ever seen where you see them ordering a differential, as they call it, okay, short for differential white blood count. And if you don't know House, that TV show, basically he's a doctor that gets cases when nobody else can figure out what's wrong with the individual. So they gather information, they run tests, okay? They're always trying to figure out if it's a virus, a bacteria, something else. And just like I said, about every episode, one of the things they say is let's get a differential or let's get a white blood cell count. And that's what they're doing is, seeing what's wrong by counting the white blood cells. Now, the opposite of leukocytosis is leukopenia, which is where they have abnormally low levels of white blood cells. And that would never be a beneficial thing. Okay, this means your immune system is compromised, can be caused by things like radiation, shock, and chemotherapeutic agents, so chemotherapy. So, not that I expect you to know the table, just know what is all involved here. All right, that's it for white blood cells, at least for now. Like I said, keep telling you this. We'll talk more about them in our next lecture when we talk more about immunity, especially those lymphocytes. Let's talk now about platelets. Platelets also go by the fancier term thrombocytes, which I've mentioned earlier, are not cells, but small fragments of marrow cells called megakaryocytes. Megakaryocytes grow really big, and then they kind of just splinter into pieces, and those splintered pieces become platelets. And as you can see, platelets do a lot for us. Okay, they secrete vasoconstrictors, chemicals that help cause constriction of broken blood vessels. So that happens like when we first start bleeding, when a blood vessel's been broken open. They can form what's called a platelet plug, which helps seal small breaks in injured blood vessels. Talk a little bit more about that here in a second, but I always tell students think of that like what you see in like TV and movies where, like a pipe is dripping or leaking and they put a piece of bubble gum over it. Not a very long-term solution, but it might be sufficient enough to get the person by for a short amount of time. They secrete procoagulants, which promote blood clotting. The word coagulation is just a fancy term for blood clotting, or the process of blood clotting or forming a blood clot. So procoagulants promote blood clotting. They also help initiate the formation of a clot, or the destruction of a clot. They form what's called a clot dissolving enzyme that dissolves blood clots that have outlived their usefulness, something that's very important because we don't want clots just hanging out and breaking free and getting into places they shouldn't. They secrete chemicals that attract neutrophils and monocytes to sites of inflammation. That's that chemotaxis stuff I was talking about, where they help chemically attract those various white blood cells to places that are in trouble. They can actually phagocytize and destroy bacteria and secrete growth factors that stimulate mitosis and fibroblasts in smooth muscle, which is something that helps a blood vessel repair itself after it's been damaged. Formation of platelets is called thrombopoiesis. So we saw erythropoiesis earlier, production of white blood or red blood cells. Here, this is the formation of platelets, or their fancy term, thrombocytes. So that's where that term comes from. Okay, kind of the same thing. We start with stem cells, myeloid stem cells that differentiate into megakaryocytes, carry receptors for the hormone thrombopoietin. TPO is a hormone that stimulates the formation of platelets. Produced by the liver when our platelet concentration in the blood decreases below normal levels. That triggers those megakaryocytes to develop into huge cells known as megakaryocytes, which stay in the bone marrow. And then, as I mentioned earlier, they kind of just start breaking apart. The inner folding of its plasma membrane will divide the cytoplasm into small compartments. And as a result, cytoplasm breaks up along these lines of weakness into tiny fragments, entering the bloodstream to become functional platelets. Platelets, though, only have a really short lifespan of about a week, 5 to 9 days, before they age and die off and must be removed by macrophages in our spleen and liver. So those are the formed elements in the blood, and to summarize them, I've included a table from your textbook that I'm not going to really spend any time talking about, since we just spent the last 40 minutes or so talking about them. All right, let's talk about hemostasis, which is a sequence of responses that stops bleeding. Now, keep in mind, it's important to visualize that the blood in our body is not just found in every crevice and every place. Okay, our blood should stay inside of your blood vessels. So when you are bleeding, that means a blood vessel has broken open and the blood is spilling out, whether that's what we can see because we've got like a cut and the blood's coming out of the body, or if it's like leaking into a cavity, what you would commonly call like internal bleeding. Okay, and you can't see that, but you might be aware that it's actually happening. Okay, when a blood vessel becomes damaged, the hemostatic response must be quick, localized to the region of damage, and carefully controlled in order to be effective. There are 3 mechanisms that we use to reduce blood loss, the first of which is called vascular spasm, a prompt vasoconstriction or tightening of a broken blood vessel to prevent further blood loss. So this should be pretty common sense. If a blood vessel is broken open, do we want to continue to send a lot of blood in and through it? No, just like if you have a leak in a pipe, you wouldn't want to continue to let water run through it, right? Okay, so you're going to turn the valve to shut that flow off. That's what vascular spasm basically is. Okay. Now, tissue damage can stimulate the smooth muscle of the vascular bed and attempt to decrease its blood flow. So when the tissues are damaged, they can help trigger this. And this effect can last for several minutes to several hours. Now, since there's 3 things listed over here on the side of the page, it would make sense that 1, 2, 3 over here on the right side would be associated with those 3 things, but that's not actually the case. 1, 2, 3 here is actually showing you number 2 over here, the formation of what's called a platelet plug. Okay. Earlier I said that the inner lining of a healthy blood vessel should be very resistant to stuff sticking to it, because it's coated with what we call prostacyclin that acts to repel things like our platelets sticking to it. Okay. But when a blood vessel is damaged, collagen fibers become exposed, which give those platelets a place to anchor and stick. Okay. The mass of platelets that initially forms, as you can see, we eventually see several of them collecting together. It's called a platelet plug. And this is going to be enough to maybe reduce or stop minor bleeding. Not significant bleeding, though. Okay. Once again, I was just saying this is like putting a piece of bubble gum over a leaky pipe, not a permanent solution by any means. Okay. Now, as a few of those platelets begin collecting, we see it especially here in step 2, they start releasing several chemicals that help promote more and more of those platelets congregating. Those include ADP, serotonin, and thromboxane A2. ADP, adenosine diphosphate, we've talked about before, helps attract more platelets to the area. Okay. Serotonin is a prominent vasoconstrictor, and so is thromboxane A2. So these 2 help with the vascular spasm aspect of things to cause that vasoconstriction in that broken blood vessel to occur. Okay. ADP and thromboxane A2 also help with attracting the platelets. So platelet aggregation means, and that's what we're really seeing right here in figure 3. Now, the bottom figure is showing you actually what takes place last, the most complicated thing, which is coagulation, the process of clotting your blood, where we convert the plasma protein fibrinogen into fibrin, which is what we see this yellowy, stringy stuff is. a sticky protein that adheres to the walls of the vessel. With fibrin, the resulting mass of cells and platelets seals the broken blood vessel. So essentially what we end up with is just a clog, like what can happen to your drain if you get something down there, like a whole bunch of hair or too much toilet paper or something like that, if you're thinking like in your toilet. Okay. And that's what seals the broken blood vessel, and that's more of a more permanent thing. But I really shouldn't even say permanent, because this isn't going to stay in there forever. We want to remove clots once we've got everything fixed up, but it's, I guess, more permanent than platelet plugs are. So how do we get to that in formation of the fibrin and stuff stuck to it? Okay. Well, it's a pretty complicated series of events where we use what we call clotting factors, which includes calcium, several inactive enzymes formed by hepatocytes released in the blood, and various molecules associated with platelets or released by damaged tissues. Okay. Now, the formation of a clot is a complex cascade of enzymatic reactions where each clotting factor is like a domino knocking down the next. Okay. Many molecules are activated in the next one in a fixed sequence. Okay, and there's two pathways, what we call the extrinsic pathway or mechanism, as I have it listed here, and the intrinsic mechanism or pathway. Okay, but as you can see, it doesn't really matter which pathway or mechanism is activated. The end result is still the same, and that's the formation of what's known as prothrombinase, which is what we show right there. Okay. The difference is kind of essentially where it all begins. In the case of the extrinsic mechanism or pathway, it's initiated by clotting factors released by damaged blood vessels and perivascular tissue, which is tissue in close proximity to, but also outside the vessel. So in the case of extrinsic, basically think of it as like outside the actual blood. Whereas intrinsic, it's using clotting factors in or in direct contact with the blood itself. Damage to the endothelial lining of a vessel can activate coagulation factors present in plasma, which then triggers cascade of events, resulting in the formation of prothrombinase. So it doesn't really matter, and that's fine. There's actually another pathway that, well, I'm just gonna shut up because I don't want to confuse you. But prothrombinase is an enzyme that takes prothrombin, hence the name prothrombinase, and converts it into thrombin. And thrombin is the enzyme that takes the fibrinogen, which we mentioned earlier, is one of those three major plasma proteins, chops it up into the tinier fibrin threads. And as you can see, the fibrin threads is the sticky protein that then the red blood cells and everything sticks to, and that's just going to gum up the whole works and clog the artery or the blood vessel completely. Now, unfortunately, there are situations where we might experience an inappropriate blood clot formation. And, I've talked about this already, but typically the prostacyclin-coated endothelial lining of blood vessels is going to prevent aggregation of our platelets. We call this ability to repel those platelets, platelet repulsion. But in conditions like atherosclerosis, that, what we see down here in the bottom right, illustrates an artery that has allowed stuff to start sticking to it, and therefore the lumen of that blood vessel is not nearly as open, and blood flow is diminished tremendously. And we talked just briefly about atherosclerosis on the last lecture. A lot of that is due to plaque, which damages the inner lining of the blood vessel and takes away that platelet repulsion. Okay. Chemicals known as antithrombins are going to delay, suppress, or prevent blood clotting by inactivating thrombin and thus preventing thrombin from converting fibrinogen into fibrin. Okay, we call them antithrombins because they stop the action of thrombin. Okay, thrombin, as we just learned, is essential to get to the final stage. And if you can't get to the final stage, you can't form a blood clot. Okay. What we would commonly call chemicals known as antithrombins are blood thinners. Okay. It's a very common misconception for people to think a blood thinner makes your blood like super watery, right? Because that's what it sounds like it does. That's not at all what it does. A blood thinner like heparin, which you can get naturally thanks to those mast cells and basophils, or you can get synthetic heparin. Like if you watch TV shows, movies, you might hear them say, put that person on a heparin drip. Okay. Or what you might also know is that aspirin can be described as a blood thinner. Okay. What they do is that they are an antithrombin. They prevent the formation of a blood clot. They reduce the likelihood that a blood clot will form, thus reduces the chance of a thrombus formation, which is just a fancy term for a blood clot. Now, great thing about aspirin is it prevents not only that, but it also prevents the formation and synthesis of thromboxane A2, which is going to affect several other things. It's very common for people to take, an aspirin a day when they're at risk for heart attacks. I don't know. I've never heard that one. Other clotting issues include hemophilia. Never heard of hemophilia before? Hemophilia is a deficiency of any of the clotting factors, thus preventing the coagulation cascade. So essentially, it's like, you think like putting a row of dominoes together, you knock each domino down. If you pull out one of those dominoes, then the whole domino row doesn't fall down, right? So if you're missing one of those dominoes, you can't get to this end goal, right? Okay. So people that have hemophilia are extremely at risk for bleeding to the point where they are, in severe danger. Okay. Even a small cut can bleed and bleed and bleed and bleed and bleed. Okay. It is not a common condition. It's actually way more common in men and boys. It's a genetic condition. Typically, the reason it's more common in men is because it's carried on the X chromosome, and men only have one X chromosome, and we get it from mom. For a female to have hemophilia, they would actually have to have it on both their X chromosomes. So they'd have to have a mom that was at least a carrier for it and a dad that had it. And that's why you don't see it very often. It was actually really common, I believe, in the Russian royal family a long time ago. That was because they didn't marry outside the family. So it was like cousins and whatnot getting married. When you don't mix genetics, things like that can happen. But it is a situation, fortunately, that some people have to live with. Thrombosis is the abnormal clotting of blood in an unbroken blood vessel. That would be a bad thing. I mentioned earlier, but a thrombus is the fancy term for a clot. It can grow very large and thus can obstruct blood flow. And if, unfortunately, it breaks free, it does happen. It is called an embolus, which can get lodged in a small artery and block blood flow. And if you've ever heard of the phrase pulmonary embolism, it's a big time killer. And in fact, this is what a lot of people can die from, like post-surgery. Okay, sometimes simple surgeries can be deadly because of blood clotting and a embolus breaking free, going somewhere, causing strokes and stuff like that. That's why, like when a person has surgery, they have to wear like TED hose, those like tights afterwards. They do a lot of different things with like the legs. So yeah, absolutely. All right, we're gonna stop here. This is a perfect spot to stop as we are basically out of time anyway, but we'll start with blood typing on Tuesday. We should be good to finish things up. Got too much trouble. We've only got, a few slides left, and then we'll move on into our lymphatic and immune lecture. It really ties things in that we had and we'll continue to talk about related to the blood. All right. Have a great weekend.